Hello everyone, my name is Priscilla Chamenyi. I am a junior data scientist for global communities at the Laboratory of Data Science at Pedro University, Fort Wayne. Welcome once again to this series of video lecture on unsupervised learning and algorithms under unsupervised learning. In the last video, we looked at k-means and PCA. In today's video, we are going to be talking about hierarchical clustering and spectral clustering. Hierarchical clustering is a type of unsupervised learning that groups data points into a hierarchy of clusters. It builds a tree-like structure that represents the hierarchical relationships between the clusters. Unlike other clustering techniques like k-means and DB-scan, it does not require the number of clusters to be specified in advance. There are two main types of hierarchical clustering, which is known as agglomerative and divisive. In the next slide, we are going to be looking at the two types of hierarchical clustering. So the first kind is the agglomerative clustering. It starts with each data point as its own cluster, and then it iteratively merges the closest pair of clusters. For the divisive clustering, it starts with all data points in a single cluster, and then it repeatedly splits the clusters. For both clustering types, merging or splitting continues until a desired number of clusters or until the desired stopping criterion is met. Now, the agglomerative clustering is the most common type of hierarchical clustering, hence is the commonly used type of clustering. Because it's the most common used type, that is why I focused on that type of clustering. And so moving forward, anything we do is going to be based on the agglomerative type of clustering. So this algorithm is for the agglomerative clustering type. As I already said, it begins with n observations and a measure such as the Euclidean distance. It treats each observation as its own cluster. And then the next step is that it examines all pairwise intercluster dissimilarities among the clusters and identify the pair of clusters that are least similar, that is, most similar. It fuses these two clusters. The dissimilarity between these two clusters indicates the height in the dendrogram at which the fusion should be placed. Now, there are four different methods that are commonly used to measure similarity. These are the measures. We have the single linkage measure, and that measures the shortest distance between points in two clusters. And then we have the complete linkage, and that measures the longest distance between points in two clusters. Then we have the average linkage, which measures the average distance between points in two clusters. Lastly, we have the Warts method, and it measures the increase in variance for the clusters being merged. The Euclidean distance is the most common metric used to calculate these similarity measures. However, other matrices such as the Manhattan distance are also used sometimes. Now, we move on to look at some advantages and disadvantages of the hierarchical clustering. For advantages, it allows for a flexible and intuitive way to explore and visualize the clustering structure of the data. The dendrogram also provides a clear visual presentation of hierarchical relationships. It is also capable of handling data sets with non-convex shape, which is a common limitation of other clustering algorithms like k-means, which assumes a convex cluster shapes all the time. Its disadvantages, however, is that it is sensitive to the scale of the data. It can be slow and memory intensive as it requires calculating and storing the distance metrics. The choice of linkage method significantly impacts clustering results. And sometimes you do not know which linkage method will be optimal for your data set. Now we are going to take a look at an example code in R. The data set we are using is the NTI 60 data set. It is a list available in the ISRL package in R. It consists of 6,830 gene expression measurements on 64 cancer cell lines. It contains, as we see here, a data frame denoted by NTI 60 $ data, and that contains the gene expression measurements for the 64 cancer cell lines. It also contains a vector, as we can see, a NTI 60 $ labs, and that lists the cancer types for each cell line. The objective is to determine whether there are groups or clusters based on their gene expression measurements that align with distinct types of cancer. So here we use we get the data set from R, and then we perform the hierarchical clustering. Take notice that we computed the distance matrix, which we said was Euclidean. That is the most common type. And then we looked at all the types of linkages. So we did a single linkage, the complete linkage, and the average linkage. We looked at the 3 types. So this is the code in R. Now, for the outcome of the code, we see the very first output on my left-hand side is the outcome for the single linkage method. The output on my right-hand side, the output above is that of the complete linkage, and the one below is that of the average linkage method. Now, one obvious difference between across the 3 dendrograms is how the clusters are being grouped. For the single linkage, you see that the cluster on the left and the right hand side are heavy, are very weighted compared to the groupings in the middle. And then for the average linkage method, we see the same output as the single linkage. However, it's more spread out. The clusters on the left and right hand side are more spread out. And then for the complete linkage, we see a more balanced dendrogram. The clusters are evenly spread out. Now, the next clustering method we're going to be looking at is spectral clustering. This clustering is based on eigenvectors and eigenvalues of the graph Laplacian matrix. It transforms data into a lower dimensional space before clustering, and it is often used when traditional clustering algorithms like k-means or hierarchical clustering are not suitable. How does this algorithm work? Firstly, the similarity between pairs of data points is measured and used to construct the graph Laplacian matrix, and then the eigenvectors of this matrix is found, and then the eigenvectors is used to transform the entire data set into a lower dimensional space, and then lastly we apply a traditional clustering algorithm like k-means to the transformed data. Now we're going to look at the advantages and disadvantages for spectral clustering. The advantages is that it is flexible and can handle different types of data. It does not make any assumptions about the shape of clusters, is less sensitive to noise, and lastly, it is powerful for clustering non-linearly separable data. Its disadvantages is that it can be computationally expensive since we have to compute all these graph Laplacian matrices, and it is also sensitive to the choice of parameters. And lastly, the choice of similarity matrix can have a significant impact on the results. So this is an example code using spectral clustering in R. We are still using the same NTI dataset, NCI 60 dataset for this clustering method as well. This time around, we are setting our clusters to have our spectral clustering to have 4 clusters, and so we see the outcome here that we have 4 clusters from the data set. This brings us to the end of today's video lecture. This work is being supported by the Laboratory of Data Science and the Indiana Data Mine grant, and it is also being supervised by Professor Alessandro Selvitella at Purdue University, Fort Wayne. Thank you for your time and attention. Till we meet again. Bye.